Welcome to this video. In this video, we're going to discuss the Renin-Angiotensin-Aldosterone System and the ADH system, or antidiuretic hormone system, to give it its full name. So I'll begin by outlining the structure for this video. So we're going to begin by going over the basic anatomy and physiology of the kidneys, because it's important for us to have the big picture of what the kidneys are and what they are doing within the human body concretely in our minds before we then discuss these two systems. After that, what we'll do is we'll discuss the two systems in turn, beginning with the renin-angiotensin-aldosterone system and then moving on to the antidiuretic hormone system. Finally, we'll end the video by talking about 4 different diseases that represent problems with these two systems. So we're going to talk about Addison's disease and Conn syndrome, which both represent problems with the renin-angiotensin-aldosterone system, and then we're going to talk about SIADH, or syndrome of inappropriate ADH, to give it its full name, and diabetes insipidus, which both represent problems with the antidiuretic hormone system. So let's begin then by going over the basic anatomy and physiology of the kidneys. So we're going to begin by looking at the macroscopic anatomy of the kidney. Then what we'll do is we'll look at the microscopic anatomy of the kidney, the structure of a nephron, and then we'll put it together and actually understand the big picture of what the kidneys are and what they are actually doing and how they work. Now, you can learn the physiology of the kidneys in a huge amount of detail. We're not going to go over it in a huge amount of detail. We're just going to look at the big picture, because the big picture is what is essential to understand in order to understand these two systems and in order to understand the 4 diseases that we want to finish off by discussing. So let's begin then with the macroscopic anatomy of a kidney. So let's imagine that we are going to take a right kidney and we're going to chop it down in a coronal plane and have a look at what its cut surface actually looks like. And we're going to pretend that we have cut down in the perfect plane so that we also cut through the ureter. So we're going to see the cut surface of the ureter as well, so that our picture is complete. So I'll draw this here. So we're looking from the front. Now, kidneys are, of course, kidney bean shaped, which is why kidney beans are called kidney beans. And here is the ureter coming off here, like so, which will of course drain the urine that this kidney is producing down to the urinary bladder where it will be stored. So, what then is the actual macroscopic structure of a kidney? Well, the macroscopic structure of a kidney is extremely simple, and it's extremely simple to actually see when you really do see a real kidney. So if you really do have a real kidney to actually dissect and you cut it open in the cross-section, it's very easy to identify the different structures that I'm now going to draw on this picture, which is one of the nice things about renal anatomy, that the structures are easy to identify. So, the structure of the kidney is divided into 2 major portions, the cortex and the medulla. Now, cortex and medulla are Latin words that are used all over the place in anatomy to describe the structure of loads of organs. Whenever you see the word cortex, it always means the outer portion of an organ, whilst medulla means the inner portion of an organ, and the kidneys are no exception. So the cortex is going to be the outer portion of the kidney tissue, and the medulla is going to be the more inner portion of the kidney tissue. Now, something that's important to understand is that the cortex tissue in the kidney projects down into the medulla in structures known as renal columns. So even though these renal columns that I'm about to draw are in the actual medulla of the kidney, they are the same sort of tissue as the cortex tissue. So let's draw this on. So here is a renal column, and then a portion of renal cortex, another renal column. So you can see that these renal columns are projections of the cortex inwards that are within the actual medulla tissue. So they're counted as part of the medulla, even though they are the same type of tissue as the renal cortex. So here we go. So this tissue here is the renal cortex, and this represents a renal column, which is part of the renal medulla. Now, there's another key portion of tissue that is also part of the renal medulla, which is known as the renal pyramids, and they're called this because they really do look like little pyramids. So remember, we are just looking at a two-dimensional cross-section, and when you cut through a pyramid in cross-section, this is what you're going to see. But if you were to see their three-dimensional structure, they've got a more pyramid, they've got a three-dimensional pyramid shape. So these blue structures that are in between the renal columns, these are renal pyramids. Renal pyramids have another name that you may well see used, which is to call them the renal papillae, but we're going to use the term renal pyramids for most of this video. So in blue, though, there, those are the renal pyramids. Right. So, to summarise what we've seen so far, we have the renal cortex here, we have invaginations of the renal cortex, or projections of the renal cortex inwards, which are known as the renal columns, and they're part of the medulla, and then in between the renal columns we have the renal pyramids, which are also part of the medullary tissue. All of this space here is draining space, and of course this is where the urine is going to be released, and it's going to drain gradually into the ureter here. This central portion of drainage space here, this has a special name. This is known as the renal pelvis. So this area is known as the pelvis. The smaller draining areas that feed into the renal pelvis, so this is the renal pelvis, these smaller little areas where urine is going to drain into and then drain into the renal pelvis to drain into the ureter, these are known as calyces. So calyces is the plural, calyx would be the singular. So this represents a calyx. Right, and that is all there really is to learning the macroscopic anatomy of the kidney. So we have the renal cortex, the renal medulla made up of the renal columns and the renal pyramids, and then we have the drainage space inside, the renal pelvis being the big bit, and then the renal calyces being the smaller bits that drain into the renal pelvis. And then, of course, you have the ureter, into which the urine from the renal pelvis is going to drain. So, macroscopic anatomy complete, and as I said earlier, if you take a real kidney and you really do take a cross-section through it, you do see this. It's very easy to identify these different structures. So, what we now want to discuss is the microscopic anatomy of the kidneys. What actually is this stuff? This is what you see if you look at it, but what is it actually made of? And how do those things that it's made of actually work? So, the microscopic structures within the kidneys are known as nephrons, singular nephron. And we're now going to have a look at the structure of a single nephron, and then what we'll do is show how the nephron sits on this picture, and then we'll discuss the basic functioning of a nephron and how this is actually going to perform the role of the kidneys. So, revision then of the structure of the nephron. So remember, the nephron is a very fancy, very complicated tube, effectively. And fluid from the blood is going to come into the tube at the start. It's going to make its way through the tube. Most of the fluid is going to be resorbed. And then the final tiny percentage of the fluid that makes it to the end of the tube, that is going to be urine. So, let's start by having a look at the beginning of the tube. So the beginning of the tube is a structure known as Bowmans Capsule, and this is the area where the beginning fluid is actually formed, and it's going to come from the bloodstream. So this is representing Bowmans Capsule, which is a structure that you're going to have at the start of every single nephron. Now, you will see that it looks effectively like a claw. Of course, we're just seeing a two-dimensional picture of it. If it was three-dimensional, you can imagine that if you rotated this round and turned it into a three-dimensional structure, it's going to be like the outside of a sphere with a hole at one side. And in this space, in the inside, this is where the capillaries are going to be placed, and from these capillaries is going to come the fluid that is going to actually make its way through the nephron. So inside of that space are going to be lots of capillaries, and I'm going to draw these capillaries in now, and this collection of capillaries that is in that space is known as a glomerulus. So here we go. So here comes an arteriole, which is now going to split into loads of little capillaries that are going to come in the inside here. So I'm drawing these like so. And I maybe have chosen a little bit of a too thick pen to draw that, but I think it gets the basic message across. So here comes a tiny little blood vessel known as the afferent arteriole. It splits into loads of capillaries, and I've tried to show 3 of those there, like so, and then they reconverge to form an efferent arteriole. Remember, afferent means coming into, efferent means going away. And this collection of capillaries there, that is known as the glomerulus. So I'll just label that up. So that is a glomerulus. And those capillaries are going to give up fluid that is going to come into the Bowmans capsule here and make its way through the nephron. And the fluid that comes out of the bloodstream and enters the nephron is known as glomerular filtrate, which for short we'll just abbreviate to GF. So you're going to have glomerular filtrate coming out of the glomerulus and into the space of the Bowmans capsule. Now, important to understand is that, of course, the blood contains loads of cells, red blood cells, white blood cells, platelets. Those do not get filtered. They stay in the capillaries. They do not become part of the glomerular filtrate that enters the nephrons. Also, the blood contains lots of very big proteins, such as albumin. That's the classic example. There's a huge amount of albumin in your blood. Albumin is a very big protein that carries all sorts of molecules around the bloodstream. So, for instance, fatty acids bind to albumin to be transported around the bloodstream, and loads of other things bind to albumin as well to be transported around the bloodstream. So it's a very important carrier protein. That does not get filtered into the glomerular filtrate. It's far too big. However, what does get filtered is water. Of course, the blood is, there is a huge amount of water that makes up the blood. That's what keeps it as a fluid. So water gets filtered, along with the tiny little electrolytes that are also dissolved within water. And the major electrolytes that we have dissolved within the blood plasma are sodium, potassium, chloride, and bicarbonate. And because this is quite an important concept, I'm actually just going to spend a little bit of time discussing this. So let's get some interesting colour. So we'll use the symbols for sodium. So sodium, potassium, chloride, and bicarbonate. These are the main electrolytes, which means charged particles that are dissolved within the fluid of the blood. And indeed, not just the fluid of the blood, but also the fluid of the extracellular fluid, and indeed the fluid of the intracellular fluid as well. Now, in the blood, the concentrations of these different electrolytes are roughly, sodium has a concentration of about 135 millimolar, potassium has a concentration of roughly 5 millimolar, and I'm using these numbers just because it makes it all work out perfectly. But these aren't the exact numbers. These are rough estimates of what the concentration will be in the fluid of the blood. So potassium has a concentration of roughly about 5 millimolar. Chloride has a concentration of roughly around 110 millimolar, and bicarbonate will give that roughly a concentration of around 30 millimolar. These numbers are approximately right, but I've chosen these values to use because, as you can see, it makes it work perfectly. So we have our positively charged ions here, and together they add up to 140 millimolar, and we've got our negatively charged ions here, and together they also add up to 140 millimolar, and that's nice because that means that everything's balanced. We've got an equal amount of positive charge and negative charge, so we've got ions and counter-ions, which is the way it should be. We do not want the blood to be charged, either positive or negative. You need to have an equal number of positive and negative ions dissolved in it. So, to summarise what we've just been talking about, so the fluid that makes up the blood serum, this contains electrolytes, dissolved charged particles, and the 4 main ones are sodium, potassium, chloride, and bicarbonate, and we've been through the concentrations that these different electrolytes are present in within the blood serum. So, when the water from the blood is filtered and produces glomerular filtrate, the electrolytes will come with it. So the glomerular filtrate will contain these concentrations of these major electrolytes. Okay, so that's something that is within glomerular filtrate. Other things that are going to be in glomerular filtrate are things like glucose, which is a very small molecule and will be filtered. Amino acids, they're very small molecules as well. The constituents of proteins, the basic building blocks that we use to build proteins, they're very small molecules that will be in the bloodstream, and they will be filtered in the glomerular filtrate. And really, really importantly, what will also be filtered is things like urea and creatinine, metabolic waste products. And of course, this is the whole function of the kidney. So this is very, very important that we discuss this as well. Metabolic waste products such as urea and creatinine. Now, urea and creatinine are the two very important ones clinically because we measure these. When we do blood tests on people, we can measure the urea and creatinine levels in their bloodstream, and we can use this to work out how well their kidneys are working. However, there are a huge number of metabolic waste products. So our body consists of a huge number of cells, and these cells are all doing fancy little chemical reactions, and these fancy little chemical reactions that are essential for life are producing metabolic waste products, loads of metabolic waste products that are generally small molecules. And two major examples are urea and creatinine, and as I've just said, these are clinically important because we actually measure them to represent the level of metabolic waste products within the bloodstream. Now, these molecules are the things that we want to actually get rid of in urine. So these are also going to be filtered by the nephron here, and they're going to end up in the glomerular filtrate. And there is another little piece of terminology that I wanted to use just then, but realised that I hadn't told you, so I'm going to add that in now. The whole structure, the Bowmans capsule along with the glomerulus, together this is known as a renal corpuscle. This whole functional unit is called a renal corpuscle. So the renal corpuscle is going to filter urea and creatinine and all the other metabolic waste products that our cells have produced and chucked into the tissue fluid and then which have ended up in the bloodstream to be filtered by the kidneys. These are all going to be filtered by renal corpuscles in the two kidneys, and they're going to end up in the glomerular filtrate. Okay, so those are important things then that are going to end up in the glomerular filtrate. We're going to have water along with the electrolytes that are dissolved in it, along with small important molecules like glucose and amino acids, and the metabolic waste products, the two major examples of which are urea and creatinine. And these are the things that we actually want to get rid of and want to end up in the final urine that we produce. Right. So let's now talk about the rest of the structure of the nephron. So after you've had Bowman's capsule, the structure continues, and you have a little convoluted tube called the proximal convoluted tubule. And we abbreviate proximal convoluted tubule down to PCT. Convoluted just means that it's wiggly, it's not straight. And as you can see, I've drawn it convoluted. So after the proximal convoluted tubule, you then have a structure known as the loop of Henle. Now the loop of Henle is a hairpin bend in the tube that loops down into the medulla tissue, into the papillary tissue or the pyramidal tissue. It loops down into the renal pyramid. So everything that we've discussed so far, the renal corpuscle and the proximal convoluted tubule, they are all present in the cortex or in a renal column, because remember, the renal columns are the same sort of tissue as makes up the cortex. So this portion of the nephron is up in the cortex or the renal column tissue. What now happens when we come into the loop of Henle is it dips down into the renal pyramid, and I'll show this on this diagram later on, but for now I'm just going to complete my picture over here. So we're going to have the tube looping down like so, and this represents the loop of Henle here. Okay, so that is the loop of Henle, and Henle is spelt like so. And for short, we'll abbreviate loop of Henle into LOH. After the loop of Henle, we come back up to the cortex tissue. So the loop of Henle loops down, but then we end up back in the cortex tissue once the loop of Henle has finished. And we have another little convoluted tubule, which is this time called the distal convoluted tubule. And for short, we'll abbreviate distal convoluted tubule down to DCT. So the proximal convoluted tubule is the one that's more proximal to the Bowmans capsule, and the distal convoluted tubule is the one that's further away from the Bowmans capsule. The distal convoluted tubule is then the end, pretty much, of the nephron. It then drains into what's called a collecting tubule, which goes through the medulla, through the pyramid, and drains into a calyx, which will then drain into the renal pelvis. So let's draw a collecting tubule here. And collecting tubules have a huge number of nephrons all draining onto them. So we'll have another nephron over here. So this is the distal convoluted tubule of another nephron also draining onto this collecting tubule. And we'll just abbreviate collecting tubule to CT. So to summarize what we've seen here, the nephron consists of the start, which is the Bowmans capsule, which has the glomerulus, this collection of capillaries here within it, and from the capillaries of the glomerulus is going to come the glomerular filtrate, which is going to enter this space inside the Bowmans capsule. This glomerular filtrate is then going to make its way through the tube that is the rest of the nephron, which consists of these three major different parts: the proximal convoluted tubule, which is in the cortex, the loop of Henle, which dips down into the renal pyramid tissue, and then the distal convoluted tubule, which is then right back up in the cortex, and then the distal convoluted tubule drains into a collecting tubule that then makes its way back through the renal pyramid and then will empty out at the bottom of a renal pyramid into a calyx. So let's just try and add this basic structure onto this picture. So we would have the Bowmans capsule there up in the cortex, then we have our proximal convoluted tubule, we have our loop of Henle like so, the distal convoluted tubule, and then we'd have a collecting tubule like so. And as I said, we'd have lots of distal convoluted tubules all draining onto the same collecting tubule because we have lots of nephrons all draining onto the same collecting tubule. So that's where nephrons are as far as the macroscopic anatomy is concerned. And this kidney, each kidney is made up of a huge number of nephrons. Approximately 1.2 million nephrons are within each of your kidneys. So you've got two kidneys, so overall about 2.4 million nephrons in total. So that is what this tissue actually consists of: millions and millions of nephrons, with, of course, a huge vascular network to create all of these glomeruli that are part of the renal corpuscles and are essential parts of the renal corpuscles. So what we now want to discuss then is the basic overview of how this actually functions to perform the role of the kidney. So big picture, what is the actual function of the kidneys? So the kidneys have lots of functions, but if you're going to just state one, the function of the kidneys is to create urine. And urine is a way of getting rid of metabolic waste products, such as urea and creatinine, but there are loads of other ones. If you did not produce urine, if I took your kidneys out of your body so that you could not produce urine, what would happen is metabolic waste products would build up and up in your blood. That is something known as uremia when that happens, i.e. when urea and creatinine and the other waste products get too high in your blood. It happens, unfortunately, in people with horrible kidney diseases where the kidneys stop working, they become uremic, and it would eventually kill you because the metabolic waste products will start to poison different tissues. So they will have effects on the brain, they will make you very confused, they might even put you into a coma, they will have massive effects on the brain. They will also have massive effects on the heart and eventually can produce cardiac arrhythmias and cardiac arrest, which is where the heart stops beating. And obviously when the heart stops beating, no blood goes to the brain and the brain dies. So uremia is very, very dangerous. We don't want that to happen. So the kidneys have this very important role of getting rid of these metabolic waste products by putting them into urine. So how do they do this? Well, the nephrons do this. So, a few little numbers then for you. We, I've already said that we have approximately 2.4 million nephrons, and each of these nephrons is continuously getting glomerular filtrate put into it, and the glomerular filtrate makes its way through the tubule, and then a tiny bit will come out the other end as urine. Some numbers. We talk about something called glomerular filtration rate, or GFR, and to keep it a nice, easy to work with number, in a healthy individual, this is around 100 milliliters per minute. Now, let me just explain this a bit more. So 100 milliliters, that's a tenth of a liter. A liter is, if you imagine a cube that is 10 centimeters by 10 centimeters by 10 centimeters, if you fill that with fluid, that's a liter. So take a tenth of that, that's 100 milliliters. And 100 milliliters of glomerular filtrate is going to be produced every minute. But that is by all 2.4 million nephrons. So each nephron is only going to produce a tiny amount of glomerular filtrate each minute. In total, if you combine the effects of all 2.4 million, the total amount of glomerular filtrate that is produced in a minute is 100 milliliters. Now, how much of that glomerular filtrate is actually going to make its way through the entire nephron to the end? Well, again, we like nice numbers. It's approximately, usually, about 1 milliliter. So to draw a little picture to represent this, so I'll have blue. Every minute we are producing 100 milliliters of glomerular filtrate across all our nephrons. And for the basis of understanding, this picture is going to now represent all of the nephrons, not just a single nephron. So it's representing the combined efforts of all 2.4 million nephrons. As the glomerular filtrate makes its way through this tube, the job of this tube is to reabsorb most of that glomerular filtrate, and in particular, reabsorb things that are very useful to us. So I mentioned how in the glomerular filtrate there is glucose and there is amino acids. Glucose and amino acids are highly useful molecules. We don't want to pee out glucose and amino acids. That would be a total waste. So all of the glucose and all of the amino acids that are in the glomerular filtrate are going to be resorbed by the tubes of the nephron, by the proximal convoluted tubule and the loop of Henle and the distal convoluted tubule. So we're going to resorb all of those useful molecules. We're also going to resorb a huge amount of the fluid along with the electrolytes that are dissolved in it. And in total, we're going to resorb about 99% of it. So if this box represents the 100 milliliters that we're producing of glomerular filtrate every minute across all our nephrons, the amount that's actually going to make it into urine is about 1 milliliter, 1% of that. The other 99%, along with these electrolytes that are dissolved in it, so remember all of this fluid has loads of these electrolytes dissolved in it, we're going to resorb all of this fluid along with the electrolytes. And then we're just going to leave this tiny 1 milliliter of fluid with the electrolytes that are dissolved in that to actually become the urine. Now here's the clever part. All of the metabolic waste products, the urea and the creatinine, well, these two are representing the metabolic waste products that are in the entire 100 milliliters of glomerular filtrate, they are going to be concentrated down into that 1 milliliter. So that 1 milliliter that's going to come out the end, so it's going to come out here, that is going to have extremely concentrated urea and creatinine and other metabolic waste products in, because all of the metabolic waste products that were in the entire glomerular filtrate have been concentrated down, so they are not resorbed. They are concentrated down into that tiny little volume of fluid, and that is then going to produce urine. In addition, just to add a little bit further information for you, I said that albumin, this massive protein that is in the bloodstream, isn't filtered within the glomerular filtrate, and that's perfectly correct. But some smaller proteins, so for instance peptide hormones, a notable example being insulin, they are small enough to actually be filtered in the glomerular filtrate. And these small little proteins that are filtered in the glomerular filtrate, they will be taken up by the cells of the nephron tubules, and they'll be broken down into amino acids, and those amino acids will then be released back into the bloodstream. So the kidney actually plays this role in also breaking down certain peptide hormones such as insulin. Right. And that is the big picture then of what the kidneys are actually doing. They filter a large amount of fluid, then they convey this fluid along this long tube known as a nephron, and in that nephron, most of the fluid is resorbed along with the electrolytes, and the metabolic waste products that were in that fluid that we filtered are all being concentrated down into the final little volume of fluid that's actually going to become urine, so that in the urine that we do produce, we have very concentrated metabolic waste products. We also want to make sure that we absolutely resorb all of the really useful molecules to the body, such as glucose, amino acids, and also take up proteins that have been filtered and break them down back into amino acids. So that is the overall big picture of what the kidneys actually do. So we'll have a break there, and in the next video we're now going to discuss the renin-angiotensin-aldosterone system and the antidiuretic hormone system.